Here we go.
Looks fantastic. So Markus, stage is yours, and maybe you can introduce yourself with a few sentences and then start with your presentation.
Thank you very much, Adinj, for the invitation and giving me the chance today to present our recent work on super resolution expansion microscopy. So a few words about myself. I'm a trained chemist with a special focus already in my PhD on spectroscopy and dye photophysics. Later on, I had a chair position in physics department for laser physics before I finally, 11 years ago, moved to Würzburg University to the biology department. So I'm mixed off between physicists, chemists, and medical biologist, so to speak. Now I'm located at the Department of Biotechnology and Biophysics at the University of Würzburg. This is at the Biocenter. And I have another shared position at the Rudolf Virchow Center, which we recently renamed into Center for Integrative and Translational Bioimaging. So I will start today with a brief introduction into the field of super resolution microscopy, really brief, 2 slides. And then I will go on to give you an overview of the current state of art of single molecule localization microscopy, of course, with a special focus on DSTOM applications, how we can visualize proteins and lipids with 20 to 30 nanometer spatial resolution, give examples. And the second part of my talk I will focus on, or I will discuss with you possibilities to achieve a structural resolution in cells and organelles of 10 to 20 nanometers or less. That is super resolution imaging in biological samples with so far unmatched resolution. This will then rather have the form of a construction site report. So I will give insights into different projects we're working on for 2 years now about to achieve this defined final goal. But let me start with a brief introduction. I mean, you're all experts, I guess, so I do not have to make many words about fluorescence. So it has been introduced by Sheenen and Stokes. They discovered the Stokes shifts working with Sheenen. Then only a few years later, von Bayer has been able to synthesize fluorescein, one of the most famous and most often used dyes we still in use today. He has been able to synthesize this organic dye from scratch. And thanks to different companies and specialists who invented and developed new dyes and labeling technologies, we can label almost any kind of cellular structure we're interested in and produce these nice images. However, as you're aware of, there is the diffraction limit. So waves are diffracted at the optical components of our microscope. And this has been written in stone as a fundamental equation by Ernst Abbe, developed at the same time as fluorescein has been synthesized for the first time so that we end up when we are doing fluorescence imaging that we act in principle like a painter, but we use a large brush, and so we smear out the image we detect on our camera. However, people have found intelligent ways and have been awarded with the Nobel Prize 2014 for chemistry, and this paved the way to bridge the gap between this atomic resolution provided on the left-hand side here by cryo-EM and electron microscopy, transmission electron microscopy in particular, and on the right-hand side, whole body imaging using, for example, positron emission tomography by fancy techniques like STAT, STORM, and structured illumination microscopy. So just a few words about single molecule localization microscopy. I will not go into details of that, but the trick behind single molecule localization microscopy, as you all know probably, is that you have to temporally control the fluorescence emission of single fluorophores. And this can be achieved by photoactivation of photo switching. The only thing you have to ensure while doing that is that while you're recording such a video as shown on the right-hand side, that the distance between these point split functions recorded, detected on your CCD camera from a single fluorophore, that these point split functions are separated at the distance larger than the diffraction barrier. Because this then enables you to localize, so to determine the center of mass of these point split functions by fitting it or approximating it to a Gaussian function. And this allows you finally to reconstruct a super resolved localization microscopy image. And the precision with which you can do that is mainly determined by photon statistics. But what paved the way in order to allow people to do these kind of super resolution microscopy experiments, this was the invention of, first of all, of photoactivatable fluorescent proteins, as introduced by Jennifer Lippincott-Schwartz and George Patterson in 2002, the PA-GFP, which allowed to activate only a sparse subset of fluorescent proteins and then do localization microscopy by reading out, detecting the signal, photobleaching, and then reactivation of the next sparse subset by irradiation with UV light. And the two papers, one of my group and the other paper of Xiaowei Zhuang's group in Harvard, which both appeared at the same time. We have been playing around with photo switching of dyes in the presence of thiol buffers, mercaptoethylamine, or mercaptoethanol, and we could demonstrate in these papers that pairs of dyes or individual dyes can be reversibly and highly reliably switched between a bright on and a very long and stable off state. And this addition of thiol of millimolar, up to 100 millimolar concentration of mercaptoethanol, for many people, it was just that, why did people work with that? Did it fall from heaven or what? No, it didn't. I mean, people who have been working in the field of single molecule spectroscopy, like Toshio Yanagida, published already in 1984 in Nature papers that addition of thiol buffers or thiols in millimolar concentrations increases the photostability. And this has been used by many groups working, especially in the field of single pair fluorescence resonance energy transfer experiments. And there people discovered strange photophysics of dyes. And this is what we took up and investigated this in more detail. So this then enables people to either use immunolabeling or toxins like phalloidine to label cellular structure and achieve localization precisions of 5 to 6 nanometers and achieve and produce these nice images with resolutions of 20, 30 nanometers. As shown here, for example, for the nuclear pore complex, which is the first paper showing that was 2012 published from us in journal Cell Science, followed then by many, many other groups that use the nuclear pore complex as a reference structure to demonstrate resolution. The beauty, however, of all this, the beauty, what makes DSTOM very easy is that you can just simply buy your antibodies commercially from different providers. And usually those antibodies are labeled with different degrees of labeling. So some of them are labeled with 1, 2, 3, 4, up to 8 fluorophores are attached usually per antibody. But just recently we could publish a paper which demonstrates, where we could demonstrate, and this was work done by Dominic and Gertie in my group, that it doesn't matter the degree of labeling for DSTOM imaging. The only thing what changes with increasing degree of labeling is that the background non-specific labeling increases. So for the higher DOL antibodies, you have more background signals because antibodies tend to bind non-specifically if there are more dyes attached to the protein. But the localization precision doesn't change, only the Fourier ring correlation, so the structural resolution slightly decreases with the degree of labeling of these antibodies. And we could also show that while those antibodies carry up to 8 dyes and they behave then in fluorescence trajectories on the left-hand side in PBS, like a single labeled antibody with a single bleaching step or 2 steps for 2 dyes attached to an antibody, or multiple bleaching steps and weird photophysics for 4 and 8 fluorophores attached to an antibody. This is also reflected in anti-bunching signals. So we do anti-bunching measurements where we can demonstrate that the light field emitted of these antibodies is really a sum of different fluorophores contributed to the light field detected. So the anti-bunching signal increases with the degree of labeling, while in switching buffer, the emission field detected is always dominated by a single fluorophore, no matter how many fluorophores are attached to your antibody. So, well, let's show or move on to a few examples. So one of the first examples in Würzburg we studied was the Synaptonemal Complex. It's a multi-protein complex, very complicated. It's formed during meiosis, and it tethers the chromosomes, the chromatid from father and mother together, and it disassembles after meiosis again. And it's a highly preserved structure among different species, and there are antibodies available commercially or non-commercially from colleagues, directed against all these different proteins that make out the synaptonemal complex. So we labeled all these proteins and did combinatorial DSTOM experiments and could finally come up with a 3D structure of the synaptonemal complex together with a group of Riccardo Benavente, and could demonstrate that it forms basically a helix in a helix structure. Let me now move on a little bit, a few examples about lipid imaging using single molecule localization microscopy. So especially ceramides play central roles in regulating the structural organization of membranes. And ceramide is the simplest sphingolipid, as shown here in the middle of this slide. And especially those ceramide-rich platforms, which have been reported in literature, as for example in the paper shown below here, they have been made responsible for that they control membrane receptor accumulation, they influence membrane curvature, vesicle formation, fusion and tracking, and that these ceramide-rich platforms should also play a role in infections. So we combined our efforts together then a few years ago with Erhard Bieberich from US, who published a paper that he could synthesize or develop antibodies directed against these long-chain ceramides, these natural ceramides. On the left-hand side, this is the paper journal Lipid Research. And we used these antibodies then and tried to visualize ceramides in fixed cells, which is absolutely challenging. And this has been done by Jan in the group at that time, which is really a challenging approach because fixation of lipids is extraordinarily complicated, as you immediately agree. In these experiments, no matter how we fixed the cells, we always found platforms, indeed platforms of the ceramides using antibodies in different cell lines, endothelial cell lines and U2R cells, Jurkat T cell lines. We always found these clusters of ceramides in the plasma membrane. Then we did dilution experiments. So we labeled the membrane with different concentrations of antibodies. But no matter how low the concentration of antibodies used was, we still saw these clusters with a diameter of about 80 nanometers, which confirms our finding that these ceramide-rich platforms might indeed be real, even though we are working with antibodies which potentially cross-link ceramides in the plasma membrane. So for all different experiments on the basal or on the apical membrane, we always found these ceramide-rich platforms with a diameter of about 80 nanometers, which contain on average 18 or so ceramide molecules. And this was further confirmed by experiments where we applied or added sphingomyelinase. It's an enzyme which is produced upon stress or also upon infectious processes. And this usually leads to a rearrangement of the plasma membrane and induction of ceramide production. And indeed, we could also see after application of sphingomyelinases that the amount of ceramides in the plasma membrane increased dramatically, and also the cluster sizes increased dramatically upon application of this enzyme. Well, other plasma membranes can also be visualized, like, for example, the glucosphingolipids, gangliosides, so-called GM1 or GB3. There's not much known about these gangliosides. These are glycolipids. They contain either a ceramide or a sphingosine as a lipid moiety. And they have been discovered in gangliosides, in neuron cells. But there's not much known about the functionality of these glycolipids, to be honest. What has been speculated and discussed in infectiology was that they contribute to infectious processes. So we joined forces together with Alexandra Schubert-Unkmeyer and again Jan Schlegel, and you see how he grew older now, a different picture on the right-hand side. And we tried to visualize the distribution of these gangliosides in the plasma membrane of endothelial cells. And we found out that you can use cholera toxin and Shiga toxin, which both bind specifically to GM1 or GB3. So we labeled them with Alexa 647, and we visualized the distribution in the plasma membrane. And as you can see here, they are quite homogeneously distributed without showing large clusters. However, if you then add bacteria, Neisseria meningitis, which express GFP, you can nicely show that there is no change in distribution for GB3, but GM1 accumulates at the entry sites or contact sites of these bacteria. And we were super happy when we saw that for the first time, but it was super challenging also to do these measurements, because you cannot measure on the basal side of the plasma membrane because there will be no bacteria. You have to measure on the apical or just at the sides of the bacteria, that you can really reproducibly and reliably visualize those bacteria and the accumulation of these GM1 gangliocytes. This brought us, or this pushed us further to look for different imaging technologies which allow us to visualize or image whole cells. And therefore, we built up a 3D lattice light sheet microscope. This was Felix, who was the constructor of this microscope, and Jan, who applied it to different examples. So we adopted the design from Eric Betzig from Genelia Farm and built exactly the same system in the lab. And this is just a study, just to demonstrate that this is, of course, ideally suited to do live cell experiments here, imaging ER dynamics and tubulin dynamics. If you have listened to the talk of Jennifer a few months ago, I think it was a few months ago, I'm sure you have seen pretty much better videos of these dynamics and the interactions between ER and tubulin. However, you can also use it to, now this video is not running. Oh God. Okay, but you can also use lattice light sheet to do 3D D-storm imaging. And this allows you, I don't know why the videos are not running now. They did run before, but they don't now. So no matter, this allows you finally to visualize the distribution also of, in this case, we focused that time a lot on membrane proteins. So we labeled membrane proteins, and we could then visualize the distribution of these proteins or these receptors in 3D on cells on the basal and apical membrane for different receptors. I'm super sorry, here is an example where you can really see the distribution of CD56. This is a neural cell adhesion receptor, where you see that this receptor, as it should be, it should accumulate at cell-cell contact sites. And this is exactly what this receptor is doing here. It accumulates at cell-cell contact sites. If you quantify this a little bit, you see that on the basal membrane, you have less signal than on the apical membrane, but at the interfaces, you have more than twice as many signals. This is a clear demonstration that these antibodies accumulate at cell-cell contact sites, but it also shows you that at the basal membrane antibodies, because we used antibodies, primary labeled antibodies for labeling, immunolabeling of the receptors, it looks like they have access problems to label the epitopes on the basal membrane of adherent cells. So we always measured for different membrane components, higher signal counts on the apical membrane. All right, let me now switch to the second part. So from the current state of art, what we have done in the last years to what we are currently working on. So, I mean, there have been different new technologies introduced on single molecule localization microscopy that obtain localization precisions of 1 to 3 nanometers. However, all these technologies become error prone, if we are honest, below resolutions of 25, 30 nanometers. And thus, true molecular resolution in biological specimen is still not within reach. Yeah? And this can be nicely demonstrated by these pictures of the nuclear pore complex recorded 2011 and 2020, now using fancy super high localization precision technologies. So structural resolution seems to be still very complicated to achieve. And this is mainly due to limitations in labeling efficiency, as all of you might know. So the size of the probes, antibodies, IgG with sizes of 10 to 50 nanometers, antigen accessibility. Then we introduce a linkage error, so which is the displacement between the fluorophore and the point of interest. then most of these super high resolution or localization techniques require or work only reliably with a special fluorophore, or maybe two fluorophores, like photoswitchability. And all these machines are super expensive and require expertise. So it would be very nice to have 3D multicolor fluorescence imaging with, let's say, 10, 30 nanometer spatial resolution, which is presently not available. And in order to achieve that, we thought about why not combine expansion microscopy with super resolution imaging. Just theoretically, using 10-fold expansion in combination with structured illumination microscopy should enable you to produce images with 10 to 30 nanometer spatial resolution in 3 to 4 colors using even fluorescent proteins, because the requirements for structured illumination microscopy are not that hard. On the other hand side, we have to work on labeling to reduce the linkage error and decrease the size of our labels. So there are two approaches we are following in this direction. Also construction sites currently, so that we try to use post-expansion labeling, so that we expand the sample without digesting completely the proteins. And by that, try to increase the epitope accessibility and to reduce the linkage error, because post-expansion immunolabeling, for example, reduces the linkage error by the expansion factor. Yeah. And we're working heavily on genetic code expansion to introduce unnatural amino acids that can be labeled site-specifically with a 1 nanometer sized fluorophore. So this is what is Gatti doing in my lab, mainly responsible for, and he was really pushing these things, genetic code expansion in the last few years, that we try to get rid of this. If you're using standard immunolabeling, you displace the fluorophore about 17.5 nanometers from your point of interest. And what genetic code expansion is using, it's using an amber stop codon, and you express an RNA synthase pair together with your plasmid for the protein of interest, and you introduce an unnatural amino acid. We are usually using lysine TCO, so it's this double bond, highly reactive double bond in this strained ring, which reacts efficiently with the tetrasign in a copper-free click reaction, finally. And this is what we used in the last and optimized in the last few years and published in many papers together with different groups around the globe, and introduced these unnatural amino acids, mainly in receptors on the plasma membrane, but also intracellular in microtubules, to visualize the dynamics of tubulin. Okay, so for those who are interested, there are papers on bioarchive where we use this technology to label proteins in neurons, and just recently in molecular cell for adhesion GPCRs, and with many other colleagues in adrenergic receptors, NMDA receptors, GABA receptors, and kainate receptors. Anyway, let's move on to expansion microscopy. So expansion microscopy, for those who have never heard about it, has been introduced by Ed Boyden, 2015, in a seminal science paper. But also, this expansion microscopy did not fall from heaven. I mean, people already 100 years ago in Dresden, in Germany, in the museum, there's the glassy women shown on the bottom on the right-hand side. So people have been working on clearing of tissue already 100 years ago. Yeah. And this was just the next step of clearing technologies, that you slightly change your polymers, which allows the polymer then to swell overnight in water. And by that, increase size, ideally isotropically, 4-fold, 10-fold, or using iterative expansion protocols, 20-fold. So what you have to do, you have to link your protein of interest into a polymer and then destroy the protein-protein interactions. You can either digest the proteins completely or just by denaturation to destroy these protein-protein interactions. And then the gel can swell. And an advantage which comes for free is that tissue samples become cleared simultaneously. Yeah, so ideally suited for fluorescence imaging experiments. And there are basically two protocols. So one is that you label your protein of interest before imaging with an antibody or dye-labeled antibodies, and then you expand. Many dyes do not survive this procedure because it's a radical polymerization step involved. Or the alternative step shown below is the so-called MAP protocol, or this was the first protocol that used post-gelation or post-expansion labeling, that you do not use a digestion step before expansion, but just denature the proteins. And this we investigated in more detail together with Paul Guichard and Virginie Hamel from Geneva and with Davide Gambarotto in a project on centrioles. And we found that time Fabian was doing the work in my lab that centrioles, in order to expand them isotropically and produce reliable confocal images of fourfold expanded centrioles via post-expansion labeling, you have to use very mild fixation. So all the standard protocols like XM, original protocol or MAP protocol didn't deliver or show or preserve, in other words, the molecular organization of these centrioles. Only if you use very mild fixation, we could really preserve the ultrastructure of these centrioles. Next step, because I have shown you synaptonemal complexes, we also did some comparisons. For example, here using just unexpanded synaptonemal complexes, which form these helical structures by structured elimination, in the middle then by D-storm, and on the right-hand side, a threefold post-labeling experiment with such a synaptonemal complex. And this allows you to do 3-color SIM of expanded samples and achieve thus 30 nanometer spatial resolution in large field of view, 3 colors of a multi-protein complex. And this is quite easy. There is no real expertise or expert expertise required in order to do these experiments and finally move to a structured illumination microscope and put the sample on the microscope. But what we discovered in all these experiments, and which is the message, is that using this post-expansion labeling, we discovered details of the organization of synaptonemal complexes which have never been seen by light microscopy before. And this is, for example, on the right-hand side, the fraying at the ends of these complexes of filaments, which is clearly seen, and also that this splitting of sublateral elements, like shown here, there's a splitting. It's not just 2 filaments, it's composed of several filaments, as shown here in electron micrographs. And this has never been possible to visualize by any kind of light microscopy before, not due to the limited resolution, due to the limited labeling efficiency, the accessibility of those epitopes which are shielded in those filament layers. You will only always be able to label the outlying filaments and never inside of these densely packed filaments. But if you first expand the samples and then optimize fixation steps and everything, you can really access epitopes which are not accessible before. So this is already a summary. I already told you that we reduced the linkage error by post-expansion labeling with antibodies, by the expansion factor about. We achieve 30 nanometer spatial resolutions in 3 color on a structured illumination microscope if we only expand 3 to 4-fold. And we have an improved labeling density. Yeah? And this provides finally enhanced ultrastructural resolution. So the question came up, can we do that with lipids? And we have been working together with Jürgen Seibel from the organic chemistry department who provided us with different functionalized ceramides and other sphingolipids, which can be clicked, so labeled with dyes by click chemistry after metabolic incorporation into cells. So we just fed cells with these sphingolipids and finally click a dye to visualize the lipid distribution in these cells. And we thought about if we, for example, attach not only an azide is for clicking of a dye here, if we attach an additional amino group, this should enable us to fix lipids in a membrane. And in addition to fixation, it should also enable us to expand these lipids. And this was work then together with Thomas Rudelskup in the microbiology department, Tobias Kunz, and again, Jan and Ralf that time worked on this, what we later termed sphingolipid expansion microscopy. And what is shown here is just a fixation that using a ceramide which does not carry an amino group cannot be fixed. It's washed out. But the amino group carrying ceramide sphingolipid can be really fixed. and cannot be easily washed out with detergents. And we took advantage out of that and did a glutaraldehyde expansion experiment. So 4 times expansion of these phingolipids. And for comparison, we did the same experiment with a peptide introduced by Silvia Rizzoli's group, M-Cling, for membrane visualization. And this is shown here in slide orange. So M-Cling mainly stains the outer membrane, but the ceramides, they stain also intracellular membranes, also mitochondria, ER, and so on. And therefore, also those organelles or the membranes of these organelles can be expanded. And it allows also the combination with immunolabeling, for example, labeling here mitochondrial marker protein PRX3 in these 2 color images to see interactions between mitochondria in magenta here, and in this case, bacteria. So cells have been infected with chlamydia, Chlamydia trachomatis bacteria, and after 24 hours, they have been labeled. So sphingolipids have been added. So these modified, functionalized sphingolipids, and finally they have been clicked, fixed, and expanded using a standard protocol. And this allows us finally to visualize that these bacteria take up some mitochondrial membrane proteins into their own membrane, for example, which was really interesting. And this can be controversially discussed, whether this is specific or non-specific, but this can be visualized using this technology. And by using tenfold expansion protocols, for example, and combining these samples then finally with structured illumination microscopy, you should be able to easily achieve a resolution of about 10 to 20 nanometers, still having 3D options and 2, 3, 4 color options available. So this allowed us at the end of the day, using this technology to visualize or to resolve even the double membrane of this gram-negative bacteria here, for example, of this chlamydia bacteria, that here the distance between the outer and the inner membrane of these chlamydias has a distance of about 27 nanometers. Electron microscopy gives you 25 nanometers. So we are not that completely wrong. And as these structures are still circular, this also tells you it's more or less isotropic, the expansion. Yeah, it's not completely non-isotropic. But please take care. I do not claim in my talk here that expansion microscopy will always proceed isotropic. So this has to be very carefully checked with reference experiments. Yeah, for example, expanding clathrin-coated pits. This is a very good reference experiment. If you still see a round circular structure after expansion, this is a very good sign, for example. So one step further, I've shown that you can expand lipids, you can expand proteins in multi-protein complexes using post-labeling. You achieve, in combination with tenfold expansion and structured illumination microscopy, you can achieve 10, 20 nanometer resolution multicolor under ideal conditions. Can we push things further and go towards molecular resolution? So there is still other problems coming to the game. And the problem is, if you try to combine this with localization microscopy, you end up with a problem that photoswitching requires buffers. And these gels are ionic hydrogels, these polymers. So if you add an ionic buffer, a salt-containing buffer, these gels will shrink. In worst case scenario, they will shrink to the down to 1/4 again, to the original size. And there is a lot of protein and fluorophore loss during digestion and denaturation and gelation. So you have to optimize these steps in order to achieve or to enable, for example, D-storm imaging combined with expanded samples. And this we did exemplify for microtubules, which I do not have to introduce here, structure of 25 nanometer diameters. If you add primary and secondary antibodies on the sides, you end up with a diameter of about 60 nanometers. So immunostaining displaces the fluorophores about 17.5 nanometers from your point of interest. If you do a pre-labeling, so you label before and then expand 3.1-fold, and then you re-embed the gel. This is, you polymerize the gel into a second non-charged polyacrylamide gel, after expansion to stabilize the expanded gel, then your originally 4-fold expanded gel will shrink to 3.1-fold about, but this allows you then to add photoswitching buffer and to do D-storm experiments of these 3.1-fold expanded microtubules. And you end up with microtubules with a diameter of about 140 nanometers. So for comparison, the unexpanded sample with the same immunostaining, you measure a diameter of about 40 nanometers. Yeah, this is about the distance from here to here. Yeah. Okay, re-embedding works. We tested this for different labeling methods. Always pre-labeling, labeling with an oligonucleotide labeled antibody, and then after expansion, adding a complementary short dye carrying oligonucleotide and things like that. But you always will end up with diameters of far more than 100 nanometer, up to 200 nanometers if you use primary, secondary antibodies and the DNA. And we also modeled all this, and this is also all confirmed by the experimental results. So it's because of the fact if you expand 4-fold a 10 nanometer antibody, it will also be 40 nanometers displaced. The antibody is also expanded. So the only way to go is again post-labeling. And this allows you then, if you do post-labeling, so you label, so you take care how you denature your sample, and then add the antibodies after expansion and re-embedding. Then you end up with a diameter of microtubules you measure by D-storm of only 80 nanometers. And this corresponds then exactly to what you expected, that adding the antibody afterwards reduces your theoretical linkage error to about 5 nanometers. You simply have to divide the 70.5 nanometers by the expansion factor about. This is your actual linkage error at the end of the day. And as we have seen, the labeling efficiency increases because of the increased epitope accessibility. So this allows you then to visualize microtubules by D-storm imaging as hollow cylinders using a water immersion lens, because you are far away from the cover slip. There is no oil immersion, there is no turf imaging possible. You have to use epi illumination, but still can visualize microtubules as hollow cylinders with a much lower linkage error. And this allowed us finally, last slide here before I conclude, to visualize also centrioles now using re-embedding and D-storm. And we can demonstrate that these tubulin filaments here in the centriole body, which are separated by about 10 to 15 nanometers according to electron microscopy, can be visualized after 3-fold expansion. So we really achieve a structural resolution which is in the range of 10, 15 nanometers. The other way to go without using re-embedding is, for example, using a spontaneously blinking dye like this 2014 introduced HMSIR, the silicon rhodamine, which shows spontaneous blinking in water, in PBS buffer pH 8. But if you work just in pure water, the blinking behavior, the expansion factor is 4-fold without re-embedding, wonderful. But the blinking behavior of this dye just in pure water is very lousy. If you then switch to PBS, the gel shrinks again back down because PBS contains salt. The switching performance increases, but your shrinking factor, but your expansion factor is diminished. So in the moment, we think re-embedding and doing just classical D-storm microscopy works best, provided that you can preserve the ultrastructure by optimizing your expansion protocol. And this is the summary of what has been shown, that this post-expansion labeling procedure dramatically shrinks the size of your label and the displacement of your fluorophore from your structure of interest, here the microtubule, because the antibodies are not expanded during your process. So you end up, even for immunolabeling, with a displacement error of only 5 nanometers using 3.1 or 2-fold expansion. And the epitope accessibility is dramatically enhanced. So we believe that this is the way we have to push further to achieve then finally maybe true molecular resolution by light microscopy. So with this, I'm at the end. I hope that I mentioned the people on the slides who did actually the work. This is the current composition of the group and the people who left the group, and also people who are looking for jobs in the moment. If you are interested in other works, come on and visit superresolution.de, and you will get more information. And these are the people who give us the money for our research. I thank you very much again, Erdinch, for the invitation to this seminar, and you for listening, and hope to be able to answer your interesting questions. Thank you very much.
Thanks so much, Markus. Fantastic. This is amazing. So you basically resolved every structure in the cell almost.
Yeah, sure. It would be happy. It's mostly microtubules, mostly filament structures.
But now you're going into lipids. This is really good. So I will start with a few of my questions first.
Sure.
First, in the first section, you mentioned something I got a bit confused. When you have multiple labels, let's say antibodies, multiple dyes per antibody.
Yeah.
And it decreases the localization accuracy because of this distances between.
That's what I think.
So, because what I was thinking, if you have more signal, it would actually also compensate for the localization accuracy. But this distance between labels.
No, the localization. The localization. That depends on what we're talking about. I mean, a single fluorophore will be localized with a precision of, depending on photon statistics, a few nanometers. But if you have an antibody with a size of 15 or 10 nanometers, and you have different fluorophores, and this guy is localized with 5 nanometers, this with 5 nanometers, and so on, this finally ends up in a smeared out of the signal again. And this decreases your final structural resolution at the end of the game. And this doesn't matter if you're using DNA paint or whatever. As long as you are using antibodies and they are multiple labeled, this will always be limiting in your experiments.
I see. And this lipid stuff, sphingolipid expansion, super interesting. So now, what do you think the limitation is? Can we do it for every lipid, specifically for lipid classes? For instance, PIP2 expansion, PS expansion. Can we do these kind of things in the future, near future?
I'm optimistic. I'm really optimistic because actually we have been really surprised about the fact that it works. It was just a theoretical consideration. Maybe you can this way fix and cross-link lipids, and we were really surprised about the fact. But of course, it all has to be tested, and it has to be carefully tested at the end of the day. I mean, you're just adding these lipids in your experiments to your cells. And you never know how they are incorporated and how specifically they are incorporated. And in the worst case scenario, they show a completely different distribution than your endogenous lipids. It's not what I believe, but it has to be considered, at least eventually, that there might be differences. Yeah. And I mean, we did also studies with longer chain and shorter chain sphingolipids. And because labeling is performed after incorporation into the membrane, you have to use short chain ceramides, because otherwise you will not be able to efficiently label them after incorporation, because then the longer ceramide fatty acid will stick into the membrane and there is no more labeling. Yeah, so you have to work with a compromise. I mean, with these unnatural short chain ceramides, and this is seriously a critics point to attack this work, and this has been done from different sides. I know what I'm talking about.
Don't worry, Markus. We have been attacked by this. Every lipid person would know actually how difficult it is to have to preserve a native behavior of a lipid when you try.
Yeah, it's absolutely a pain in the neck. This fixation of lipids. I mean, what we, for example, I mean, I usually don't talk about it, is that with these antibodies, we see ceramide-rich platforms. If we feed cells with ceramides, we have never seen these platforms. So is there a difference between endogenous ceramides, just freshly transported to the membrane, and those fed externally? Question mark. Question mark.
Welcome to the lipid world, Markus.
Yeah.
Okay, there are many questions by the audience, so I will start. First question, Jane Xiong. Hello, a practical question. Regarding the paper, multiple labeled antibodies behave like single emitters in switching buffers. Does this extend to measurements of on-duty cycle? Because some papers report you need low dye to antibody ratio below 1 in order to calculate the duty cycle.
Yeah, that's right, in a way. If you read this paper carefully, at the end of the paper, we show that for many of these multiple labeled antibodies, there is enormous blinking going on in the first few seconds of the experiment. And during this first few seconds of the experiment, we are pretty sure that most of these fluorophores already bleach. So, and if you now consider how you start the storm experiment, you start with a very bright sample at the beginning, and then you illuminate your sample, you irradiate your sample for the first 1-2 minutes to push the fluorophores into the off states that you see the blinking, and then you start the experiment. But during these first few seconds, you already photobleach many of these fluorophores which are attached to these antibodies. So it might well be that you end up after a minute with with the majority of antibodies labeled with a single fluorophore at the end. And this is at least our explanation, because we don't see huge difference in statistics. There are no clear differences, which is what you expect for a 4-fold labeled or a single labeled antibody in blinking statistics.
Thanks, Markus. Next question is by Pablo Caravilla. He has 2 questions. First, the membrane question. You mentioned that ceramide platforms might be induced by the antibody cross-linking. Did you try FABs instead of IgGs? And if so, was the platform size the same?
No, just due to the fact that we didn't have the Fab fragments and we didn't try to produce them ourselves from the antibodies. Also, the amount of antibody provided from US was so low that we didn't consider to start to produce our own Fab experiments. But we did experiments at 4 degrees centigrade, for example, on ice as well, and using very low antibody concentrations, and still seeing these clusters with diameters of about 70 to 80 nanometers. This convinced us that time that it's real what we are seeing at the end of the day.
I mean, overall, the ceramide antibodies, ganglioside antibodies, super interesting. In the body, they definitely work.
Yeah.
I mean, and they're related to a lot of autoimmune diseases, so they must.
Yeah, it might also be due to the fact that the problem that these antibodies directed against lipids are, and that's at least my impression, that they are not directed against a single lipid. They are might be directed against lipid domains, and so they always recognize lipid domains and they label lipid domains, and not individual lipids in the plasma membrane.
Yeah, that's actually, that's definitely a possibility as well. A lot of peptides, actually, they recognize the domains, but not single lipids.
Exactly. Yeah.
All right, Pablo's next question is about expansion. How much does labeling efficiency decrease in post-labeling due to epitope digestion?
I mean, if you really use proteinase K to digest the proteins, your post-labeling efficiency will decrease to 0 for sure. Maybe I didn't understand the question completely, but I mean, we did, for example, just maybe this answers the question. We did an experiment where we did pre-labeling of microtubules with immunolabeling. Then we expanded using mild denaturation, and then we added again the same antibodies, immunolabeling, to see how broad the microtubules are in our D-storm images. And they were very small. So which tells us that post-labeling, immunolabeling outcompetes pre-immunolabeling. So we saw something like with small shoulders on the sides. I hope that this answers the question, that post-labeling can, under certain circumstances, really outperform pre-labeling.
Mm-hmm. Okay, next question. Why don't you use unnatural amino acids in expansion microscopy? Why would you prefer using antibodies?
Well, we're working on it. That's one of our construction sites, as I mentioned. I mean, this is, of course, ideal, the ideal approach, yeah? But it's, in the moment, all these natural amino acids is associated with overexpression. So you leave the endogenous side, and this is also complicated. But for sure, we're working on that, because this is, from our point of view, it might be the key.
Mm-hmm. Thanks, Markus. So next question is by Christina. In which cell structures can we observe super resolution expansion microscopy the most clear? Is there a difference in imaging on the basis of, let's say, organelle, cell membrane? What would be the gold standard?
I can't answer the question. I'm aware of a paper of Christian Eggeling's group, nice paper in Chem Biochem, where they demonstrated that different organelles are differently expanded. So at least different expansion factors. So the only advice I can give is you have to test carefully your protocol for for your target of interest. You have to optimize each step of this expansion protocol. And this is why I said a very easy control experiment, if you are interested in membrane expansion or organelle or endosome expansion, is that you use immunolabeling of endosomes or cluttering-coated pits. And if there are circular structures after expansion.
Okay, that's good. All right, next question is by Luis Evers. So did I get it right that the persistence of the protein structures in the gel is depending on the amount of NH2 groups?
I mean, the NH2 groups are required in order to, no. I mean, either you label your protein with an antibody, and then the antibody is fixed and linked into the gel, because the antibody has many amino groups. In case of these lipids, it's the amino group attached to this functionalized unnatural sphingolipid we are using. This amino group can be fixed, so cross-linked with glutaldehyde, and then glutaldehyde can be linked into a polyacrylamide gel.
Okay. Related to this, Markus, I wonder what limits us in the expansion, really? Can we expand in the ideal condition? Can we expand 50 times, 100 times?
Yes, it has been shown by Boyden, but I mean, these polymers are non-ideal as well, and this is what Boyden is working on. And just, I think this week or last week, a paper appeared in Nature Nanotechnology about a new tetragel, where he used a different chemistry to produce these gels, which has less errors, yeah, less holes. It's more homogeneous and allows you to more homogeneously link proteins of a multi-protein complex into this gel. I mean, you have to ensure that within let's say you're working with a hexamer, that each of these hexamers is linked into your polyacrylamide gel. And this is one of the problems in the moment, because these current gels cannot 100% provide or guarantee this in the moment, and therefore you will use. And these 50-fold expansion protocols, they use iterative expansion, which is you expand, then you digest a linker enzymatically or reductively, and then you polymerize the next gel, which further expands. And this way you can expand up to 50-fold. But my gut feeling is that the reproducibility decreases from expansion step to expansion step. So from our side, we are focusing a lot on these single-step 10-fold expansion protocols available in the moment.
Thanks, Marcus. The next question is by Luca. Have you tried to use nanobodies,
Yes.
single domain antibodies, to reduce the error?
Yeah, of course. We also used nanobodies mainly directed against GFP.
Mm-hmm. And does it help to reduce errors?
Sure, absolutely. I mean, sure, you see exactly what you expect. A nanobody has a size of only 3-4 nanometers, and this is exactly translated into your linkage error also in expansion microscopy.
Great. Next question is by Ege about are the biophysical properties of membrane different in the areas of membrane enriched with sphingolipids and ceramides?
Mm-hmm. Oh, well, I cannot say too much about this. It's supposed that in these rich areas that the membrane is much more flexible. And this is also the reason why people believe that these are the entry points for viruses and bacteria, because they're much more flexible. But it might also serve as an accumulation point of some receptors this way. But this is still speculation, I would say. We're currently also working on visualization of SARS-CoV-2 viruses together with lipids, and also there it looks that lipids play an important role. This is what I can say in the moment. It's not so the receptors, or maybe the receptors are important, of course, but it also it points towards the direction that lipids play a much underestimated role also in this coronaviral infections. I do not have details in the moment. I'm really sorry, but we did so many experiments the last year because time was so boring. We always came up with the same, lipids play a fundamentally important role.
Marcus, I'm going to quote you in my grant applications. Lipids play fundamental roles.
Yeah,
from now on, I'm going to quote you. I'm going to quote you for every application I have.
No, that's for sure. We have to come up with some really convincing experiments, yeah. And the field is very critical.
Yeah. Okay, while talking about lipids, maybe last questions, Marcus. Well, you're now expanding lipids and proteins, but the field of biology is probably much more interested in DNA and nuclear structure. Do you think this expansion will be applied also to solve this very dense structure of nucleus that we cannot access?
Absolutely.
This packing.
Absolutely. This is in focus of. I'm aware of 3 groups worldwide who focus their efforts into this direction. I mean, it has been the method. It has been the method of the year of Nature Methods. Spatially resolved transcriptomics. So this is exactly where to go into this direction. I mean, you can digest DNA, you can link DNA into a polymer, and by that do expansion fish of RNA or DNA and do this spatially resolved this way. I mean, there was just recently this science paper of Ed Boyden and 100 co-workers or so, where they did single-molecule DNA sequencing in expanded samples in a science paper. Even spatially resolved. So this was very impressive, super interesting to say. Yeah.
Thanks a lot, Marcus. Okay, my last question, I promise. Since I catch one of the pioneers of super resolution microscopy here, what do you think now with MinFlux, MinStead, and ideal case, you do D-storm, you approach this molecular resolution, right? D-storm with expansion. Do you think we will get almost EM details with the light microscopy? And what do you think the main bottleneck is?
The main bottleneck is still, we have the microscopes in hand. The localization, new refined localization microscopy techniques like MinFlux, they approach 1 nanometer localization precision. And this is for sure. But if you have a look on the images produced in biology so far, you see that labeling and displacement is still the bottleneck for all these high localization precision techniques. And from my point of view, many biologists are super happy with if they use tenfold expansion and combine it with standard microscopy, confocal or structured illumination microscopy, because then you achieve already 25 nanometer resolution in 3D. Yeah. And if you really aim to go for 1 nanometer or for electron microscopy, this can only be done if you use, for example, this genetic code expansion and you insert linkers site specifically, and you have a polyacrylamide gel or a polymer or a swellable gel, which allows you to link all these monomeric units into the gel for expansion. And we're not there yet. This is reality, but we might, or maybe we are there in a few years, and that's at least what we're working on together with many colleagues.
Brilliant. So I cannot thank you enough, Markus. That was really fantastic.
Thank you.
Thanks again for joining us today.
Thank you very much.
It was a pleasure. So hope to see you soon.
Hope to see you all soon, sooner or later. Bye-bye.
Bye. Bye, Markus.